Hello, alright, I'm just gonna charge up my MacBook. So, hi there guys. Hope you're all enjoying your holidays. I know I am. It's been alright. Honestly, I haven't done any revision up until today. And today, I went to bed at 2 o'clock in the morning. I woke up at 12, so I've had a solid amount of sleep, but like, my sleep schedule is messed up now. Like, seriously, it's messed up, and I thought, well, I could spend the whole day drawing and, like, playing Subnautica, but I would much rather try and force myself to do some revision so that I can get into the swing of it again, because if I end up bloody, getting back to school day one of, week 3 or 4, whatever, of GCSEs, and I end up slipping up, making some dumb mistakes, it's gonna suck. Now, today I thought we should do Science Paper 2, because that is the one that we did with he's an okay guy, but, like, in terms of science, he ain't the best. So I thought, why not go through and try and learn some of that. So I'm trying to find. I had a little sheet with all of the DNA and inheritance stuff, and I don't recall where I put it. I'll be honest, I don't recall. I have like 100,000 drawers just full of there it is. I have a giant pile of just books. My computer's taken a while to load here, so this is going to be Biology Paper 2. You guys can't actually even see that. Okay, I guess I'll write it down here. Biology Paper 2. We'll move this back a little, actually. We'll crank it up a little. There we go, okay. Biology Paper 2. And we're going to go through, there's plenty of topics to do in Biology Paper 2. You have one of my favourites, ecology. You have the DNA topic. You have homeostasis. And I believe that's it. I believe it's only 3. What I'm going to do is I'm just going to pull up the stream on my little computer, so that if I do get any questions, any people wondering what's going on, they can merrily be informed. But yeah, hope you guys are doing well. I know that I am. I just uploaded one of my favourite videos that I've done this year, well, the only video that I've properly done this year. It's called Back to the Forest. Basically what it is it's a remaster of the forest video. It's a lot more detailed, a lot more like ambient. You got some cool little Easter eggs and stuff scattered in it. Anyways, I'm just gonna add absolutely everybody now. Okay, so let's get into things. Gonna close my computer now. So, we already know a bit about this topic, okay? We know that we have, we can rub all of this out now. We already know somewhat, so let's assume here, like, we don't know anything about, so we're doing the DNA topic. I hate the homeostasis topic. I will learn it, but I cannot be asked to today. I'm too tired. So we'll just recap the DNA topic, and then we'll do a bit of ecology. So we have, of course, DNA, and that is obviously the whole ATCG thing, and basically DNA kind of, I guess, ravels together to form the chromosomes, which is mitosis. I'm not sure whether we need to know about mitosis for this topic or not. I hope we do, because I still remember it. It's that I prepare meals at the college. That's like the little acronym, but I used to remember it. But I feel like mitosis is a paper. I mean, it was literally on the paper, wasn't it? It was literally one of the questions explaining mitosis. It was only 4 marks, wasn't it? But that's 4 marks I got, so, you know. Now, we have 2 different types of genes. You have the dominant and the recessive. You may get asked what these are. I hope we do, because basically the dominant will always be expressed. And recessive will only be expressed if there are 2 of the same alleles. I watch this YouTube channel called Primer. He does a lot of like biology and statistics videos, so basically my whole thing that I love. And he does a lot of videos explaining like genes and stuff. He runs like little simulations of like society and how, you know, genes can affect everything. It's such an interesting channel. And yeah, these are the 2 important words, dominant and recessive. So recessive is normally represented as little numbers, such as A and A. Dominance is usually the big numbers, capital A, capital A. Now we can draw these funny diagrams that I think everyone hates. I don't mind them, but they can be a little bit tedious. So I believe it's male downwards, female along. So if we had a recessive male, I believe that would look like... Oh yeah, I was right, okay. So if we had, this is to show recessive, we would have like A and A, A and A. So obviously recessive genes, which means that is going to be expressed, but nothing else will be. If that makes sense. Basically, if 2 things are linked together, two of these lowercase letters, it means that it'll be expressed, but any other combination won't be expressed. So, it's normally in percentages of a quarter. So for example here, this is a 25% chance that you get this recessive disorder or this recessive trait. In terms of dominant, I believe it's quite the opposite. It's... so it's like this. Something like that, where dominant is obviously going to be a 50% chance there instead of a 25% chance. So it's weird, these little squares are a little bit annoying to do, but basically just remember that for dominant, you only need 1 to be expressed. So again, we'll do another example. Let's see, B and B, B and B. So then let's assume that's 1, that would be expressed. In fact, all of these would be expressed. So it's an 100% chance. Because look, there's a capital B in every single one. If the lowercase b was the dominant one, then it would only be a 75% chance. So I hope that makes sense. Now we need to learn about, how exactly does protein synthesis occur? Because I believe it's DNA codes for proteins, or something like that. Oh my god, what, bro, my hair's like... Why does my hair look so dark all of a sudden? Like, bro, it's been bleached or something. Opposite bleached. Anyways, so we have here our nucleus. It's normally drawn as kind of a little, like, invisible looking thing. Which links to, obviously, how mitosis occurs, where the nuclear membrane breaks down. So anyways, inside of this, we have DNA. And the DNA never leaves the nucleus. But what happens is, in order to copy, it unzips, basically. So it creates, like, 2 kind of identical strands. It unzips. Imagine it's almost like, literally a computer. You get a zip file, you unzip it. So the DNA unzips, it then moves. Hang on, I'm missing a step. No, I'm not. So it unzips, and then it moves via diffusion. You need to remember basically. In biology, a lot of the stuff you can link to either respiration or one of the movements: diffusion, osmosis, active transport. This is diffusion, and it moves because the RNA. I don't understand what I wrote here, honestly. RNA makes copy that. It makes the DNA copy. I'm no idea. So I'm missing a step here, if I'm not mistaken. But basically it moves into the ribosome. And that is how protein synthesis occurs, because basically it reads, so if we look inside the thing, it reads this DNA in threes. And each three is then joined together as a protein. So if we have 3 here, this would literally just be 1. 3 here, that will be another, and it's joined together in proteins, in a way. So that's basically that. I'm missing a step here, and it's annoying me. It's something about RNA and like messenger things. I'll go have a look at it quickly. But it basically unzips, moves to a ribosome, and it's run in threes. Let's have a look. Let's have a look. Okay, let's have a look here. So, the DNA code for protein, so the DNA itself remains in the nucleus. Oh, okay, I missed the step. RNA is copied. So basically, the DNA in the nucleus unzips, and then a copy is made, which is called RNA, which moves from the nucleus to the ribosomes, and then from there, it is obviously translated using the things. Alright, that makes a lot more sense. So carrier molecules bring specific amino acids to add to the growing protein in correct order. Not sure what that exactly refers to. I guess this is after the fact. So after the proteins are actually synthesized here, carrier molecules bring amino acids to add to the chain. Okay. So yeah, DNA structure determines the protein that's created. So it unzips, so a copy of it is made, but it's only one part, it's not the full thing. I guess that's what you need to remember. So that's protein synthesis, quite a weird one, for sure. I guess I also should mention homo and heterozygous. Homo, if you imagine like homosexual, it's 2 of the same, so that would be 2 of them, but hetero would be H and H. There are some really scummy exam questions where they'll give you like a family tree, and then they expect you to be able to like kind of work out, they expect you to be able to work out the probability of getting a certain disorder, but normally the family trees are massive in length, and they kind of don't make sense sometimes. Those questions are the ones that I dread, because I can plot stuff in one of those funny squares fine, but if it's put in a weird family tree like this, it becomes a lot more difficult to really understand. Now I'm wondering if I have any biology papers that I could go through. I probably do downstairs. but I cannot be bothered to get them. So we'll go online and we'll look. So yeah, it's number 5, homeostasis, inheritance, variation and evolution, ecology, and then key ideas. So I forgot to talk about evolution here. So you have like Charles Darwin. There was a really scummy question last year on, it was a drawing of Darwin or something, and it was like, why is he drawn to be like half monkey? And you have to be like, well, actually, it's because he believed that monkeys were like the ancestors of humans or something like that. But it kind of a really random question about Charles, so you gotta learn him, I guess. So let's go look at the things. The question papers from whenever, component paper 2. And I want to look at the standard. Alright, let's have a look at the November 2021 higher paper 2. And let's just see what we can try and answer here. I'm going to be honest, I honestly am so tired today, but okay. Figure 1 shows part of a... Okay, hang on. Name the structures inside the nucleus that contain DNA. Well, that's obviously chromosomes, because chromosomes in each one, they contain the DNA. So let me go check to make sure I didn't make some dumb mistake there in my tiredness. Chromosomes. Nice. Easy 1 mark there. I'm going to make a tally. I'm going to make a tally of every mark that I get and every mark that I don't get in this. So then I can kind of... Because I feel like a lot of people in my class share the same level of knowledge about paper 2. Because obviously we had Mr. Hooper for the whole thing. So I definitely feel like if I struggle with something, you guys will also struggle with it, and vice versa. If we're good at something, we're good at it. A lot of people hate the ecology topic. I don't mind it. It's very wordy, and that's what I like. You can really ramble about something, and you can get a lot of marks. The more technical stuff in the DNA topic and the homeostasis, that's what I don't like. Maybe you guys would agree there. Homeostasis is a lot of explaining stuff, and it's really tough. So anyways, that's 1 mark. Okay, figure 1. So again, you guys won't be able to see this, but it basically shows something, almost DNA molecule. And it says, name the part of the DNA molecule labeled X. Let me try and draw this. So it shows like A, T, C, G, A, T, like that. But then it also has kind of these weird things here with like hexagon shapes. I'm drawing them as squares so that it's quicker to draw. But it's these weird hexagon shapes. And this one here is a phosphate. What is this one here? That should be a sugar, if I'm not mistaken. Basically, DNA is almost... I think, isn't it kind of bonded, structured together? It's a DNA molecule, but it's kind of structured together using sugar and phosphate. If I'm not mistaken, that should be the answer. Yep, sugar. Nice. So yeah, sugar and phosphate are like the 2 big DNA molecules. In fact, I think that's why they call it like deox. That's what DNA stands for. It's like deoxyphosphate something acid. But like that deox bit, I believe, refers to like phosphorus and sugar. What type of substances are labelled A, C, G, and T? A, C, T, G. If I'm not mistaken, what are these, actually? Genes? Is that the wording I'm looking for? Oh, bases! I forgot that was a thing, honestly. Bases. Aha! I remember now, because the bases bond together. They don't actually, if I think that's just the way I remember it. A, T, C, G, those are the bases, and I like to say that like they bond together, because A and T are always kind of connected, and C and G are always connected. Basically, bases bond. Alright, that's a good way to remember that then. Figure 2 shows another section of a DNA model. Oh, this is a lot of diagrams. You guys might need to pull up an exam thing yourself here, because this is taking up a lot of time. That would have been, so that's another mark, and then I lost 1 mark there for something very easy there. I literally dropped a mark for forgetting the name. But that's all good. Figure 2 shows another section of DNA. 4 of the substances you named in question 01.3 are not labelled. What the hell does this mean? 4 of the substances you named in question are not labelled. Label each of these substances. Oh, this is literally just connecting them together. So A and T, C and G. That's literally what you do when you get a mark. That was easy. What is happening to the DNA in part Z of figure 2? So that is showing, it shows that over time, they're kind of becoming separate. Over time, they're kind of becoming separate. And it looks as if the whole structure itself is kind of pulling away. That's unzipping, right? But I don't have a terminology for that. Replication, I'm going to say. Yep, it's replication. So the DNA unzips, but it's basically replicating. That's another mark. Okay, next question. A gene is a length of DNA. What type of substance does a gene code for? It's either amino acids or proteins. Genes code for proteins, right? Or do amino... Yeah, it's protein. Okay. Overthinking myself, that's how I always slip up. This is how I always slip up marks. I always end up overthinking it. But there we go. 5 marks already. That is 5% of this paper for literally just putting some words down. DNA codes. In fact, I'm going to start to write some stuff here so we can recap back on this. DNA code for protein. We have bases. And the unzipping is replication. See, this is what I like to do. I like to go through the past papers and just collect little bits of information so that I can use them. Most human body cells contain 6 times 10 to the power of minus 12 grams of DNA. What mass of DNA will a sperm cell contain? Okay, sperm cells, and this is something to include because this could be in a thing, are gametes, I believe. Basically sex cells. And they always contain half the amount of chromosomes because basically when they fertilize, they get the other half from the female gamete. So it contains half so that when they're connected, I guess, it's a whole. So in other words, if you have 6 times 10 to the power of minus 12 grams, you would have 3 times 10 to the power of minus 12 grams in a sperm cell. So they contain half. They contain half the amount of chromosomes. That was a nice and easy mark. Hope you guys also got that. What is the name of a type of cell division that produces sperm cells? This is... I don't know how you pronounce it. It's meiosis. Meiosis. I don't know. Meiosis. I'm going to call it meiosis because then the E is exaggerated so you guys know what I'm on about. Meiosis, if I'm not mistaken, is the exact same as mitosis, but there's an extra step. It's good, but it's good if I can teach you guys this now because it will teach me as well. Okay, how does it work? Is it the same as mitosis? Okay, so yeah, we know this for chromosomes replicate. We know this. Remember, I prepare meals at the college. So okay, we'll draw the full thing just so that you guys can get it. Okay, so the chromosomes replicate. That's step number 1. Oh, and then shit changes totally. Okay, shit, this is different. Similar chromosomes pair up. So similar chromosomes pair up. Then the sections of the DNA swap. So I guess there's no real way to visualize this, so I'll just put swap. Sections swap. The first cell division occurs, so the chromosome pairs separate, and then the second division happens, so that all you're left with are kind of like wiggly worms. Okay, I hope that made kind of sense. So to begin with, we know this, they replicate. That's the terminology you have to use. The chromosomes replicate. Then, we have they pair together. That's kind of it. Then, the sections of DNA swap. And then, they replicate. They divide. The first cell division occurs, the chromosome pairs separate. So I guess that would be better to just put separate. And then finally, they separate again. Alright, we need to learn how to remember this. RPS SS. RPSSS. Or ps. I don't know. They replicate, they pair, they swap, they separate, and separate. ERPS. If you guys unironically remember that could ERPS. That could help you get a lot of marks. And hey, look at that, we learned that in a minute. Oh, okay, right, we're moving on now from the inheritance stuff, so I want you guys to remember that. ERPs, so they replicate, first and foremost, the chromosomes replicate, they pair together, they swap, the DNA kind of does a little dance, it swaps, well, that's how I'll remember that. And then the chromosomes separate due to division, and then they separate again due to further division. The DNA codes for proteins, when it unzips, it replicates. We got it, guys. That was a very, very nice first bit. That was 7 marks out of 8 that I got there. A student investigated the effect of temperature on the decay of milk. So he first sets up the apparatus, as shown, with the milk at 20 degrees. He records the pH over 5 days and repeats with another batch of fresh milk at 25 degrees. How could the student keep milk at a constant temperature for 5 days? Ooh, Put it in a fridge? Or like a closed environment? I need to think carefully here because the exam board is scummy. Keep it in a closed environment. That's what I'm going to say. Oh no, water bath. It's a water bath. How would that work though? Because surely water loses temperature over time. Okay, there's something right away that'll not only help you with paper 2, but like chemistry as well. Water bath keeps temperature. Put it in a bath and preserve the temperature. Give one variable the student should keep constant. Volume of milk. Because if it starts to go down, you're going to get kind of weird results, aren't you? Yep, volume of milk or type of milk. Perfect. So there we go. 1 mark there, 1 mark there. We're winning so far. That's 8 out of 10. Oh, plot a graph. And it's 4 marks. And, just as I was actually about to say, this is probably going to be a curve graph. If it looks like a curve, it is a curve. That's what the teachers always say, and that's what a lot of people, including myself, often forget or panic about. That would have probably gotten me 3 marks, I'll be honest with you. I might have dropped 1 mark for not doing a curve. So yeah, 2 marks for plotting the points, correct scale and axis, but if you join it point to point with straight lines, you won't get it. It has to be a straight curve. Nice. All right, that would have been a nicer question. The data you plotted in question 02.3 were obtained at 20 degrees Celsius. Sketch a line on figure 4 to show the result you would expect at 25 degrees. Label this line 25 degrees. Oh, okay. So if it's at a higher temperature, it's going to decay faster. Right? Because the enzymes are working at a higher optimum temperature, which means it'll decay faster. And if we look here, over time the pH increases. In the first couple of days, it doesn't do anything, but then over time, it slowly decreases, or I guess increases in pH. This time around, I think it would do it faster. Would it not? Similar shape drawn to left of 20 degree line. Same start pH. I'm so confused by what that means. I don't think I would have gotten any marks there because I can't visualize that. That's hard to visualize that. Students line are best fit for the students plot of Yeah, I don't think I would have gotten any there because I can't, that's insanely hard to visualize. I get how that works. It's not giving me a clear explanation here. The mark scheme is being a bit weird. I'm sure that it would decrease. So the pH does that faster. Would I have gotten that there? I'm gonna be very strict because I always am when I'm doing past papers and give myself no marks for that. No marks. Again, the reason I'm strict is because I think worst case scenario. Like in my biology paper, I said worst case scenario, mid-50s to low 60s. So maybe 55 marks to maybe 60 marks. That's like the ballpark average of what I got. Best case scenario, it might be like 65 marks, but Worst case scenario, mid-50s. That's what I think. Which means, and that is only about 20% of the GCSE, right? 27%. Which means we really well and truly... Hang on, let me just look at the map. Let me have a look at the grade boundaries. Right, I'm looking at the November 2022, which I think are resets. I need biology. Biology, higher. A 9 was 132 marks. So let's assume that because a lot of people thought this paper was nice, let's just round that up very strictly to 140 for a 9. And then so a 7 is 104. Let's round that up to 110 for a 7. Right, in the last paper that I got roughly, lowest case scenario, a 55. That means in order for me to get a 7, I need to do identical to this as that. In other words, I need to get the exact same marks. Let's say best case scenario here, it's 110 for a grade 7, and I got 60. I only need to get half marks next paper. So I literally can drop maybe this paper 40 marks, which is great. But still not the best, but you know, it is what it is. It is acceptable. So I've already dropped 5 marks. I can only lose another 35. So we're only about like 10% through the paper so far. Human body temperature is controlled within very narrow limits. Scientists investigated the effect of drinking ice-cold water on internal body temperature and the rate of sweating. So the method they used is they sit a guy down, kept at a constant temperature in a room, measure their internal body temperature near the brain, measure their rate of sweating. After 20 minutes of this, they give a person half a litre of ice-cold water. They continue to measure their internal body temperature and sweating. Give a reason why the person should not move during the investigation. Well, when you move, it generates heat. Your muscle, it increases heart rate, could you not say? Which would increase the rate body temperature. Let's look at a proper answer for that there. Yep, extra movement would release extra heat. That's perfect. That I would have gotten a mark for basically parroting what the mark scheme said there. We are 12 out of 50 marks there for getting a 7. Let's assume that I need a 60 though. Let's be really strict on ourselves. So a 60, that is still 10%. 10% there. Some really nice questions on this one. Okay, so this is the scientist results. It is a graph. And it starts off, so that's the body temperature. It ducks down and then it goes back up. So this is the point where they drink the ice cold water. This is their temperature. And basically, we can determine that once they drink the water, their body temperature decreases, but then it increases shortly after. The same for the rate of sweating. After about 10 minutes, they begin to sweat less, but over time it goes back to 0. What is the person's normal body temperature? Seems to be 37.4. That's the bit there that it's constant at, but it always returns to. So I'm going to say 37.4. That would make the most sense. Yep. There we go. A nice and easy mark there just for using my eyes. We're getting closer, guys. The results show that when the ice cold water was drunk, the temperature near the brain decreased. Why did the temperature near the brain decrease? This is only 2 marks, but come on. 2 marks and we'll be a quarter there. Why the temperature near the brain? Why did it decrease? Okay. 2 marks, 2 points. We could say that... I can't even think. Why did the temperature near the brain decrease when he drank ice cold water? Well, because the water was cold. So cold energy was... I can't think of the words here. Shit. Oh, it's to do with blood. Your blood is cooled when it goes into your mouth, because when you drink cold water, your blood is cooled, and the cooled... It's blood! It's blood! Okay. Shit. That could have been 2 easy marks. Think about it. Here, I'll go drink some water now to demonstrate. Right? Look, we got water. I drink it. And you can kind of feel it. You can kind of feel it kind of disperse a little. You can also... It leaves a lingering, when you drink water, it kind of leaves a lingering kind of taste in your mouth. I guess that's almost for blood in a sense. Like, the temperature of a blood. That's why I could probably, in the next 10 minutes, I'll feel my face getting a little bit colder. But that'll be why. It's to do with blood. Because it's homeostasis! Why did I think of that? Homeostasis is blood. It's all the blood stuff. Shit. Why didn't I think of that, bro? That's actually annoying. That's 2 marks easily that I bloody dropped there. Bollocks. Okay, now we know that homeostasis is blood. Because you obviously have... I don't even remember the names of them. The one where your blood vessels like dilate, and where they like become narrow. I don't remember the name of it, but that is obviously happens in your blood, and homeostasis happens with glucose in your blood. Bollocks, man! 2 marks for that. Oh shit! I'm spoiling the answers to myself. Yes, it's a thermoregulatory sensor. Yes, that is it. That is it. Damn. Honestly, why did I forget that, bro? That's the only bit of homeostasis that I pay attention to. Thermoregulatory sensor in the brain responds to the decrease in temperature. How does the thermoregulatory sensor send information to sweat glands through the nerves? Via nerves / neurons, via nerve impulses. Okay. I'm assuming I would have got that there. Again, I'm being incredibly strict on myself here. If I even, like, slip up a tiny bit of a mark scheme, I will not give myself a mark. Because I'm assuming worst case scenario for our exams here. But yeah, those are them. Welcome to the stream, by the way. I was raging over homeostasis here. I'm just going through a past paper from November 2022. Which means, yes, I will need to delete the stream once it's done, because I legally can't show past papers on stream. I believe, anyways. The rate of sweating changes between 24 minutes and 36. Explain how this change helps to maintain the person's normal body temperature. Well, sweat releases, I guess it just releases the thermal energy. Um, it removes excess. Again, I know what it does, but getting the terminology, the mark scheme terminology. Wait, hang on. I didn't even look at the thing. The rate of sweating Oh, it decreases. Okay, but I wouldn't have got that anyways. Less sweating occurs because Oh, it's ice cold water, isn't it? So when you have cold water in your blood is cooled, so you stop sweating as much, because otherwise, if you keep losing, it's gonna get even colder. That's formula regulatory stuff. So basically, when your blood is cold, less sweating, and I believe the other blood vessels like dilate, and then they no, not dilate, the other one. I can't remember it, bloody hell. And when they're warm, more sweating. Your blood vessels. Oh, contract and dilate. That's it. Yes. During exercise, the skin appears red. What causes the skin to appear red? Dilation of blood vessels in the skin. There we go, nice. So that would have been maybe 2 marks there, because I literally just misread the question. Obviously, in an exam, I would have looked at the graph and realised it decreased, but there I just jumped into the conclusion. Oh, it's biodecomposition. Decay occurs in a compost heap. Cool. Describe how it's a 6-mark question. Shit. Describe how microorganisms in the layers of soil help to recycle chemicals in the dead plants, and how the chemicals are used again by living plants. Okay. I'll be honest, a couple of months ago I looked at this mark scheme, so I already know some of the things. So how do the microorganisms in the layers of soil help recycle the chemicals in the dead plants? Well, you could say that they digest the dead plant matter. I know that might not be a point, but I'll get into a point here. And basically, they use this matter, they use the glucose. Complete guess here. It's dead plants though, isn't it? They might not have any glucose. They use something to respire aerobically. That has to be a mark. The living plants themselves get the CO2 released from the respiration, and they can use that to make glucose to grow. The chemicals. What other chemicals are in plants that can be used? I mean, literally just glucose. Starch? But if they're dead, they won't have any starch. Mineral ions? This is honestly a tough one here, because again, we have not learnt this. Digestion. Oh, hang on. Carbon dioxide taken in by the leaves by diffusion. You just had... Oh my god, you literally could just state... photosynthesis and would have gotten 5 marks. Are you taking the piss? You literally just had to say CO2 taken in by leaves, by the stomata. CO2 is used to make, used in photosynthesis for glucose, which is then used to help a plant grow. Man, so seriously, you could have gotten like 4 marks there. So I would have actually gotten a level 1 response there, because all I did was state the facts, simply state them, but not include relevance, I don't think. So I probably would have gotten 3 marks there. Let me have a look at what happens in the microorganisms. They digest large molecules into small ones. They respire. They produce CO2. They release mineral ions. Okay, I would have gotten at least 2 marks for that then. The fact that they digest, so I guess you should just say the large molecules become like smaller ones. They use these larger molecules, they digest them, so they respire to release CO2. I would have gotten 3 marks there. I'll be dead strict on myself and say I only got 3 marks there. That's 18 out of 60 that we need. So that would have been a really nice question had I just gone into a tad more detail with everything there. Had I just kind of explained the process of photosynthesis, which I obviously know. So there you go, guys. For the 6 mark questions, keep on using connectives. Keep on saying and, by, but. So this happens by the process of, via this, because of this, which does this. I'm going to probably end the stream in a minute, but we'll do a couple more questions. Ooh, abiotic and biotic factors, okay. Nitrates in the soil, are they abiotic or biotic? If I'm not mistaken, abiotic features... I honestly need to check here. So biotic are features that animals cause, and abiotic is everything else. So nitrates being in the soil would be abiotic, because it's not something that a random rabbit did. They didn't put nitrates in the soil. Rabbits eating the plant would be biotic. Shading by a building, abiotic. Soil pH would be abiotic, because it's out of the control, I guess. Temperature, abiotic. Trampling by people would be biotic. Oh, it's a 2. Is it a 2 mark? Oh, it's 8 marks for quadrat stuff. Shit. The student estimates the number of daisy plants growing on the lawn. The student places a quadrat at 10 different positions on the lawn. The student counts a number. How should the student decide where to place the quadrat? A random method in order to get a variety of results. Maybe? Randomness. Oh, no, okay, randomness was the reason why. Grid and coordinates. Or literally just say random... You could literally just say random coordinates and get 2 marks. Damn. The mean number of daisies in each quadrat is 6. Calculate the number of daisies on the lawn. Okay. This is a math question. And I don't like math, but we'll give it a shot. We'll give it a shot. So that was two easy marks there. So we have this. We have what appears to be a paved area. So we'll have to take this out of our results. 6, 4, 3, 6, 10, and it makes it very clear that it's a triangle. It's a 90-degree triangle, a right-angle triangle. So the mean of each daisy is 6, and the quadrats are 50 centimeters by 50 centimeters, which means in a square, it would be 1 meter, 1 meter squared, right? Yes, that is right. 1 meter squared. So we need to calculate the volume, the area of this, which would be 16. So the total area is 160 divided by 2. So the total area of the area is this. We need to minus this, which would be 18. So 18 minus 18. I'll just do the method just to show that we know how to do it. 62. So this total area is 62 centimeters squared. Now we know that per each quadrat there are 6, which means there will be 24 per meter squared of 24 daisies per meter squared of quadrats. So if there's 24 per meter squared, we do 62 times 24. Which will get us 1,488 daisies. Please be right, for the love of God. Yes! We got it. No! I wouldn't have got it because I didn't round to 3 significant figures. No! I would have gotten 5 marks for that. But I did do it right though. That was the method. Calculate the total area, calculate how many per meter squared, and then just multiply them together. I would have gotten 5 marks for that. Bollocks, man. So much working out too. You guys have to admit. But anyways, that is 25 marks. And we are currently on... How many more questions are on this paper? Maybe... So we were on question 5 point something. There are about 10 questions total. So we were halfway through with only half of the possible marks. Let's look back at what we need to do to improve. Okay, number 1 is we need to read the questions. Alright, we need to read them carefully. That said round to 3 significant figures, and I didn't. I didn't round it at all. Literally would have gotten full marks for just adding 2. That's 1 mark. We now know that homeostasis, it's the blood. So link everything in homeostasis to the blood. If you want to keep the temperature of something constant, use a water bath. We now know bases. We know that obviously DNA codes for proteins. We know RPSSS. That's the way I remember. Meiosis. That's all I'm doing today. I hope you guys learned some stuff. I did. A lot, actually. I now know to read the bloody questions. We will continue this past paper tomorrow. Maybe. Or maybe later today. But 25 marks out of 12. So 37. 25 out of 37. Let me do the math here. 25 over 37. That is 67% right. And I would have literally gotten 1 mark for rounding and a mark for drawing a line of best fit. So that would have got me 75% right. Right, well anyway, it's 25. If we divide that by 37, and then we multiply that by 100, if I am able to maintain that level of accuracy, I would in fact get a grade 7. I would get 67 total marks. We'll round that down though and we'll say 65. Which is incredible because that's great. So if I can just learn to take a tad more time reading the questions, things like that, and if I can just remember 1 or 2 of the little cheekier methods that I've done today, if I can get the first 5 questions totally right, which is, I mean, 65% right, that's pretty damn good. for our first try at a paper, which we haven't done in ages. But honestly, yeah, I would argue that was pretty good. So you guys will have to let me know if you were answering in the background or whatever, how well you think you did there. Remember, the point of these streams is that it's like a community as well. If you guys had a problem with anything there, if any of you guys didn't understand my way of explaining something, take your own time and do some bloody like research or something, you know? Take your time to do, you know, to Google something, go on BBC Bite Size, spend 5 minutes learning it, and it could save your ass.